Hello everyone. So in our last lecture we were talking about blood. We spoke about the blood in like the composition of blood. We spoke about composition like what is there in the blood, RBC, WBC, plasma. We spoke about the functions, the different kinds of functions of blood, and then we finally spoke about the physical characteristic, and then finally about blood plasma, the components of blood plasma, what's the function of that then. And then what we started to discuss was the formed elements, which are the cells. So blood has three types of cells like we discussed: RBCs, WBCs, and which is red blood cells, white blood cells, and the platelets. And they have their own characteristics, right? So what we will discuss in this lecture is basically the RBCs, or three erythrocytes. Erythrocytes and the RBCs are the same thing, okay? Red blood cells or erythrocytes. So erythrocytes obviously are very small in diameter, about 7.5. This is called micrometer, okay? 7.5 micrometer. Micrometer is even less than millimeter, 1000 part of a millimeter. So you can imagine how small one RBC is. And what they do, they help us contribute to gas transport. And when we were talking about gas transport, we're talking about oxygen, firstly, because that's our respiratory gas. We cannot live without oxygen. And then CO2, which is a metabolic waste. Now, this particular cell has a biconcave disc shape, means like this. Cave number 1 and cave number 2 looks like this, okay? It is anucleate, means there is no nucleus and there are no organelles either. So this is filled with, all it has is basically filled with a molecule called hemoglobin that is required for gas transport, because that's the only job red blood cells mainly does. So RBC's diameters are, we have some blood vessels actually which are even smaller than RBC. So RBC is slightly bigger than that. So we will, when we talk about blood vessels, we'll talk about them. And RBC also have a protein called spectrin. There could be other proteins obviously, but there is a one special protein called plasma membrane protein. The plasma membrane protein, I mean the membrane protein of RBC, which is called spectrin, which actually gives the flexibility to its shape, helps the RBC to stay flexible in its shape, which is biconcave again. So this is how it looks like. So again here, like you can see, this is cave number 1 and cave number 2. So this is cut in half and the whole diameter is 7.5 micrometer, really small, right? And this, because it has this extra dip in its shape, that actually gives it extra area for containing, for gas exchange, right? So hemoglobin makes actually 97% of cell volume, and then there's some little bit of water in there too. Now RBC doesn't have its mitochondria, so it requires ATP, but that is completely anaerobically produced. But the funny thing is, they don't consume any oxygen while they are working, but they only transport that oxygen. They don't consume anything. All right. So these are, like I said, dedicated for respiratory gas transport. Firstly, oxygen, slightly can be a bit of carbon dioxide also. So what is there in RBC? So the main component is hemoglobin. Okay, so what is this hemoglobin? Hemoglobin is the molecule that really binds to oxygen, right? So we do have a volume or value of hemoglobin. That, I mean, we need a required amount of hemoglobin in our blood. Okay, in your blood lab, you should be testing. You do have an optional test, I think, to do with hemoglobin. So what happens here in hemoglobin, males have approximately 13 to 18 grams per 100 ml. Males should have hemoglobin like that, and females 12 to 16. So this value needs to be maintained. If it's lower than this value, then that could be a problem, that could be a disease actually. Now, hemoglobin contains a red heme pigment, first of all, and then bound to globin. So that's why it's called hemoglobin, heme and globin. What is this globin? Globin is a big protein. It is a protein made up of 4 polypeptide chains, 2 alpha, 2 beta. You have read about this. It's a quaternary state of protein, right? 4 chains. So 1, 2, 3, and 4, and each has its own heme. So this is the heme component. Each chain will have its own heme component. That's where the oxygen binds. At the center of it, you will see that there is iron attached to that, and this is actually where the oxygen really binds. So therefore, each hemoglobin molecule, since it has four chains, four globin chains, and each globin chain has four hemes with ions, so therefore each hemoglobin can transport four oxygen. Okay, this is something that you will have to remember, must. So if a hemoglobin has four oxygen, it is carrying four oxygen. Not necessarily it will always carry four oxygen. If it is, then it is called as 100% saturated. 100% saturated. If the blood, suppose, is carrying 3 out of 4, then we will call that hemoglobin molecule is 75% saturated, right? Or if it is only carrying 2 on 4, then we will say it is 50% saturated. So depending on how much oxygen it is carrying, we can determine the saturation of hemoglobin. So each RBC contains 250 million hemoglobin molecules. So you can imagine how many are there. Now, when the hemoglobin is loaded with oxygen, for example, it is coming right out of lungs, because lungs is the one that pumps oxygen into the blood. We breathe oxygen from air, right? And that's when we call it as oxyhemoglobin. It is ruby red in color. But suppose the blood is coming out of tissues, and now tissues have already done their metabolic functions, liver, stomach, heart, all of them. So therefore that blood will be loaded with uh, less hemoglobin, right? So I mean less oxygen in that hemoglobin. So then we will call that as deoxyhemoglobin or reduced hemoglobin, and that will be dark red. Okay. And if there is carbon dioxide also can attach to hemoglobin. In that case, we will call it as carbinohemoglobin. And 20% of carbon dioxide in blood actually binds to hemoglobin, and then that produces carbaminohemoglobin. Although too much of concentration of carbaminohemoglobin is not a very good idea for our body to have. Now, how are these erythrocytes produced? They are, first of all blood cells are produced by a mechanism called hematopoiesis. Poiesis means synthesis, and hemato means it is related to blood. So, formation of all blood cells means hematopoiesis. Now, when we are specifically talking about RBCs, erythrocytes, then we will call it as erythropoiesis, the formation of blood cells. It takes about 15 days. This is the whole process. So we do have stem cells for these also. Now the stem cells, even though it's a erythropoiesis, that will have nucleus, right? So it has stem cell. First of all, it will form a committed cell called porerythroblast. It will go through different developmental pathway. In one particular phase, it will eject the nucleus, because the final product will not have a nucleus, and then finally it will form a reticulocyte, then from erythrocytes. So this process takes about 15 days. Typically, this erythropoiesis happens in red bone marrow within our bone, and it is generally bones of our axial skeletal girdles or proximal epiphysis will actually have this red bone marrow. Of humerus, if you remember from 208, humerus and femur will be filled with the proximal epiphysis, right? The epiphysis, or the area that is close to your body, epiphysis, that part will actually is filled up with red bone marrow, and there is a majority of our like RBCs are produced. It also happens in some of our girdles and then many other bones of axial skeletons as well. All right, so when it comes to regulation and requirements, we cannot have too few RBCs. If that happens, then we will end up in a condition called hypoxia. Hypoxia means less oxygen. Now since uh, RBCs carry oxygen, so if we have less RBC, so naturally our body will be less in oxygen. We will call that as hypoxia. Is too much RBC good for us? No, that because RBC is a very thick molecule. The globin, this protein molecule that's in there in RBC, is very thick. It makes RBC very thick. So we cannot have too many RBC as well. Then that will increase the blood's thickness, which is the viscosity. Viscosity means thickness. Okay, so we cannot have too much thick blood either, then it will have trouble in passing through our blood vessels. And there is a number that is like produced in our number of RBC that is produced in our blood. So we typically generate approximately 2 million RBCs in a second. Now, how RBC will be produced and destroyed? It has got two things. Two things really control it. First of all, there is a hormone that controls RBC production, and then we have to supplement certain things from our diet. So let's see. First, hormonal control, and the hormone is basically called erythropoietin, EPO. This hormone is produced from our kidney. Okay. Remember, we learned about this little bit in the previous chapter. So erythropoietin is actually produced by kidney. So suppose we have normal blood oxygen level, so then that's fine. But if we have hypoxia, means inadequate oxygen is being delivered in our body, maybe because we have decreased RBC count, so that is, or we have decreased amount of hemoglobin, something, or maybe there is decreased availability of oxygen. Any of these conditions is enough to trigger the kidney to make erythropoietin more. So when, so these are the stimulations, and when this happens, erythropoietin is made by kidney, and then that stimulates the red bone marrow in the bones to create more and more RBCs, so that the oxygen carrying capacity in our blood is again goes back to normal. So that is the hormonal control. So erythropoietin, EPO, is the hormone that controls RBC production. Now, what is then the dietary requirement? Our dietary requirement is actually through food. So we need to supplement, we need to have enough amount of, first of all, amino acid, lipid, and carbohydrates, because amino acid, again, the building block of proteins, and lipids, carbohydrates are also part of many. For example, the erythropoiesis itself would require lipid and carbohydrate energy, and lipids also will provide that. So all of that metabolic energy requirement is coming from these molecules, and amino acids, it is what it makes the protein, and hemoglobin is made up of a big protein called globin, right? So therefore we need all of these to be substituted from our food. Another very very important thing is iron, which we have to supplement from our diet. If we are low in iron, then we will have to supplement with medicine maybe also, right? 60% of our body's iron is found in hemoglobin, and the rest, or it can be in liver, or it can be stored in spleen or in bone marrow. But it free iron molecules, or free ions in blood is not good. So that will be toxic. It has to be stored somewhere, bone marrow or hemoglobin or liver, somewhere. It cannot be freely available. If it is, then that's toxic. We also need vitamin B12 and folic acid, that is required for DNA synthesis, for the rapidly dividing cells, that we require for the generation of, when we are producing more RBCs, then we require vitamin B12 and folic acid, because they play a very important role in cell division, including RBC cell division during regeneration, during the generation of RBC, because RBC itself, after it is matured, it's not going to divide, only during the generation process of RBC. Now, what's the lifespan? Is it RBC doesn't live very long. It only lives for 100 to 120 days, 3 to 4 months. They are anucleates. They cannot synthesize new proteins. They cannot grow, cannot divide. Old RBCs, they become fragile, and the hemoglobin eventually basically degenerates. Sometimes RBC can get trapped into circulatory channels, especially in clean. They need to be cleaned up. And if there is some old RBC, then that is can be engulfed by macrophages. Macrophages are special types of cells that takes care of any waste product that is generated in our body. So it can take care of old dying RBCs as well. So RBC, when we are talking about RBC breakdown, so we are talking about three things to be broken down, right? Heme, the iron, and then the globin. So the three need to be broken down. So iron actually binds to ferritin or hemosiderin, and it is stored for reuse. It is stored in some tissue for reuse. Hemoglobin is not the hemoglobin, only the heme component. It is degraded into yellow pigment, because it's a pigment, so it is degraded into yellow pigment, bilirubin, and then liver really secretes out through bile that bilirubin. So heme actually gets out of the body. But globin, which is a you do not need to go into much details of this. Just know that it is secreted as bilirubin. The heme is secreted as bilirubin through bile, and that happens through intestine. I sorry, through liver. Secreted, the bilirubin actually secreted from liver. So the globin itself, it is metabolized because globin is a big protein, so it can be metabolized into smaller amino acids, and then that is released into circulation. We can use those amino acids again. Now there are some major kind of erythrocyte disorders can happen. For example, I'm sure you've heard about anemia or polycythemia. Now, what is ammonia? Means the blood is not carrying enough oxygen. Okay, and this is itself is a symptom. It's not really a disease. And you may have some other disease, and as a result of that, this symptoms might appear. Now, the reasons could be many more because, for example, due to some disease, maybe not enough RBCs are produced, or RBCs are, even if it's produced, they too much are getting destroyed, or maybe accident has happened and too much blood loss have happened suddenly. So all this can cause actually into anemia. So anemia is itself will not happen just like that. It has to happen for a reason, for some other disease. There are several kind of anemia. For example, hemorrhagic anemia, which can happen from accidents, like suddenly you're losing blood. Uh, you have to be given blood again. Blood has to be replaced. Like if it's a chronic hemorrhagic anemia, for example, you are continuously losing blood, and that can happen if a person has ulcer. Okay, then that blood loss again has to be stopped. Iron deficiency, the person is low in iron because iron is part of the heme, and that is basically that component is carrying the oxygen. So therefore iron deficiency anemia, that will give rise to anemia, and anemia is low oxygen carrying capacity, so here the iron is less. Pernicious anemia, this is an autoimmune disease. Why? Because autoimmune means it's basically attacks our own or affects our own immune system. So in pernicious anemia, actually, it destroys a mucosal lining of stomach. So it affects the stomach's inside lining, mucosal lining, and that mucosa is an important part of the immune system as well. So when that lining is destroyed, so immune system is affected, so that's why it is autoimmune disease. Renal anemia, it means we will lack EPO, and we just read about that EPO is required for production of erythropoiesis, right, or production of erythrocytes, so it is required for erythropoiesis. So therefore renal anemia means we are lacking in EPO. And then finally, aplastic anemia means the red marrow itself, where erythropoiesis again happens, that is being destroyed. Now, this can happen with maybe due to drugs or due to chemicals, radiation. Sometimes cancer patients actually can suffer through this because they has to go through radiation. They have to go through radiation, or maybe some virus can also cause this kind of thing. So it's called aplastic anemia. If there is too many RBCs are destroyed, that is called premature lysis of RBC. That is called as hemolytic anemia. This can be caused by incompatible transfusion. Again, we will know about blood groups later. Now, incompatible transfusion means you have a blood type and you're given another blood type. Maybe someone had some blood loss due to some accident or something, and that person is given another a blood that is not matching with that person's blood. So I have an accident and I have like my blood group is A positive, and I've been given like B positive blood. Suddenly that is not compatible. A and B, they are not compatible each other, so that will have that is called as incompatible transfusion. In that case, the RBCs will be undergo lysis. Lysis means breakdown. So also can happen due to some abnormality in hemoglobin. Due to thalassemia, this is means one globin chain is absent, so naturally the RBC is affected. Sickle cell anemia, the RBC looks like this. It looks like a half moon, a crescent shape. So naturally, because the shape is damaged, so oxygen levels are low. Prevalent in African malarial bent and other descendant. And polycythemia, this actually means excessive RBC. That's why poly. So the blood is too thick, too much viscous. All right, let's quickly talk about these concept checks. So RBCs are efficient oxygen transport cells of the following characteristics. Which of the major contributor is a significant contributor to its oxygen carrying capacity? Well, RBC does not have mitochondria, but it has nothing to do with its oxygen carrying capacity. RBCs don't divide. That has again nothing to do with the oxygen carrying capacity. Is the shape that it affects. So therefore, the right answer will be C, the biconcave. We discussed this. Each hemoglobin can transport how many number of oxygen atoms, right? So you will go back here and then go back in the structure over here, and then figure out how many actually it can, how many oxygen atoms can be carried out. All right. So a patient, the next question is, a patient low iron levels which experience which of these following symptoms? Low iron level, right? So increased white blood cells, iron and white blood cells have nothing to do with each other, so we can easily opt out the white blood cells options. Either it will be increase in energy level or increase in fatigue. So energy level will definitely not increase. Why? Because we are low in iron level. So low in iron level means you have low oxygen or low heme part, right? So therefore that person will be tired, increase in fatigue. Now here I would like to point out that your case study from this chapter is actually based on anemia. So before you attempt that, make sure you are reading this part of the chapter very well, right? All right, then the next thing we'll talk about is leukocytes, which are the WBCs. So we will actually learn about their classes, structure, classification, functions, how are they produced, what are the disorders, and then also about platelets. So leukocytes and WBCs. Now the major difference between these two, that leukocytes have nuclei and some organelles as well. These are cells actually. And although it makes up less than 1% of the blood volume, approximately we should have 4,800 to 10,800 WBC per microliter of blood. If we have less than that, then we might have other problems, and we'll talk about that. And there are different kind of functions that leukocytes are involved with. Includes the first major function is defense, defense against disease. Leukocytes are major part of our immune system. Okay, so from here I'll conclude this particular lecture, and in part 3 we will actually more talk about leukocytes and platelets, and keep working on the concept checks. So when we're done with this chapter, like chapter 16, you will submit the concept check. All right, so we'll see you then.